So, hello everyone, and thank you for the organization for the opportunity to present our work. I am Ghislain Fievet, a PhD student. Today I'll be presenting algorithms that we developed belonging to the class known as adversarial attacks, specifically applied to a type of data known as single-cell transcriptomic data. Adversarial attacks are a class of algorithms that make small changes to a classifier input in order to provoke big perturbation in the model's prediction. On the top left, you can see an image of a panda correctly classified as a panda by a classifier. After adding a specifically designed small noise, the model now predicts it is a gibbon. Or the opposite, making big changes to the input while keeping the same output. Below, random noise images have been modified so that the model believes that they are an armadillo, a bubble, a centipede, and so on, even though they look nothing like an armadillo, a bubble, or a centipede. Adversarial attacks have been studied on many types of data. For example, on image classification, they've been tested on anomaly detection classifiers on X-ray images and on histopathology slides. They also have been studied on text data, like sentiment classifiers that changes its prediction when a few words are replaced by some synonyms. And on time series, like electrocardiogram recordings, where small changes can make models detect fake anomalies. Single-cell transcriptomics is a sequencing technique that measures RNA expression in each individual cell. Typically, this results in a matrix with one column per cell and one row per gene representing cell expression levels. There are classifiers designed for this type of data, which can predict the cell type of each cell based on its gene expression profile. And we represent the cells in 2D with the dimension reduction algorithm. What we did is develop specific adversarial attack methods tailored to single-cell data and their classifiers. These methods test how robust classifiers are when gene expression values are altered. There are two main existing adversarial attacks methods used in pictures or in single-cell data. The fast gradient sign methods, or FGSM, which adds a small change to the input in the direction of the gradient of the model in order to make it modify the originally predicted class. Projected gradient descent, or PGD, does the same, but applies several small changes step by step in an iterative process. These two methods have already been studied on single-cell data, but they only work with neural networks and where the model weights are accessible to compute and use the gradient. However, most cell-type classifiers in the literature use other machine learning algorithms, not neural networks. That's why we developed new methods that can be applied to any type of classifier. Our goal was to make adversarial attacks usable in a more general and realistic settings. We developed three main methods and implemented them in our package available on Bioconductor. The general workflow is we start from an RNA expression matrix annotated with cell types, and our methods try to switch the predicted type of a whole cluster by modifying the gene expression. The first method, we called single gene, is the simplest. It tries to change the classifier's prediction by modifying only one gene by using a dichotomic algorithm to reduce computing time. In the paper, we tested 5 classifiers, 3 from the literature and 2 in-house, and found that 4 out of 5 were vulnerable. The predicted cell types of a cluster could be switched to another cell type by modifying only one gene. The second, max change, does the opposite. It modifies as many genes as possible while keeping the same predicted cell type, still with a dichotomic algorithm. And finally, CGD for cluster gradient descent, which estimates a kind of gradient using finite differences and uses it to slightly change several genes in an iterative process until the cluster's classification switches to another one. Now I'll show some example results of these methods. Here is an example from the max change method. On the left, you can see a heat map of the most important genes permitting to distinguish the different cell types. These data sets come from a breast tumor metastasized to the axilla, and we annotated it with the cheetah classifier. Columns are genes, rows are cells, and on the right, there's a color bar showing the cell types. For example, the cell we attack is in green, which are fibroblasts. On the left, there is the original data. We can clearly see the expression signatures of this green cluster. On the right is the result after the classifier. The signature is completely gone, but the classifier still predicts the cells as fibroblasts. Here is an example using the CGD method. Just like in the previous slide, the left heatmap shows the original data, which we annotated with the SC annotate R classifier. We are still attacking the green cluster, which corresponds to kidney epithelial cells. To run CGD, we need to provide two parameters, alpha and epsilon, which control the strength of the modifications. The slide shows how these parameters affect the result. In the middle, with small values, alpha equals epsilon equals 0.5, we need to modify 15 genes to alter the classification. On the right, with alpha equal epsilon equal 1, only 2 genes need to be modified, but the changes are much more visible. We can clearly see 2 blue lines on the right. So why is this important? These cell type classifiers could be used in future clinical applications. Traditional ways of evaluating classifiers rely on existing annotated datasets, and we check if the model gives the same labels as the ground truth. But this approach has limits, especially for rare or unexpected situations. So we want an extra safety layer by testing how classifiers behave in unnatural or artificial scenarios. If a classifier changes its prediction just because one gene is modified, that's a potential issue, or at least something we want to be aware of. So I'd like to thank the NGR Lab for hosting me, and especially Professor Brozeus, Dr. Ergalan, as well as Professor Mer for their collaboration on this project. I also would like to add that I'm available for a postdoc starting March. Feel free to find me for a chat. And you can also come see our poster today on another part of my PhD, using AI to detect features that are interesting, but that are not detectable by classical statistical tests. I would like to thank the organization and thank you for your attention.
So much for your talk. We have time for one really short question.
Really short, thank you. About this one gene, then everything got destroyed. That's very worrisome. Is there something in those genes that make these things vulnerable? I imagine that it's maybe the genes that reflects the best the separation of the clusters.
Yes, that is this phenomenon. The genes are biologically relevant, but if we are realistic, we don't want only one gene to modify the classification. But yes, they are usually biologically, except for the neural networks, when we put very aberrant values for the modification, then like half of the gene can lead to a modification of the cell type.
Thank you. Merci.
Thank you so much.
Thank you.
Thank the speaker again.